So students, please write your name. I see that many of you have done it already, so continue writing your name in the chat. Thank you, everyone. Thank you, everyone, for joining us on the third school seminar and the last one in this semester. If you're a student, yes, you wrote your name. So in the first, oh, I didn't share my screen. Or I shared the wrong screen. Just a second. So in the first, in the first two seminars of the semester, my goal was to connect between the members of the school by sharing our effort to make this significant contribution in education, science, and raising public awareness to the ocean. And I think that this goal was fully accomplished thanks to the amazing work of the speakers in this seminar. I personally was looking for the seminar each month, and as they inspired and encouraged me, I saw that this discussion helped to generate a sense of scientific community that shares the passion for both science and giving back to the public and the ocean. I do believe that actions bring hope. I also think that through these seminars, we get to know our colleagues better and learn about their career paths, which I find fascinating. So today I asked the speakers to start their talk by presenting their science and then share with us how this science got them to reach out and become active in public education and awareness about the ocean. And our first speaker is Debi Ramon. Debi is a PhD student in the marine biology department. Debi had influenced all of us, and me personally. I stopped using and buying and using disposable utensils after Debi master defense. So Debi already influenced my life. Debi grew up in California. As a teenager, she volunteered as a small local aquarium that aimed at exposing the public to local wildlife in California waters and raise awareness of the marine environment. She made Aliyah to Israel after she finished high school and did her bachelor degree in environmental sciences at the Tel Hai Academic College. She wanted to better understand the human impact on marine systems, so she joined the Marine Biology and Marine Geosciences department of our school as a master's student. In her master's, she studied microplastic accumulation in the local fish community. She is continuing to work on this subject and on heavy metal and organic pollution accumulation in her PhD, which she does with Goodman Tchernov and Professor Peter Kroot from NUI Galloway, which she met in the course that she will tell us about. Today, she will tell us about her shipboard training across the Atlantic Ocean and how it led her to becoming a team member of the We See Foundation in Eilat. So go ahead, Debi. The stage is yours.
Thank you. Thank you for a wonderful introduction. Let me get to my screen. Okay. So hello, everyone. Thank you, Swandal, for inviting me. Thank you for thinking. It's very humbling, the thought that I think that was worthy enough for a school seminar. So thank you. So today I'll be telling you all about my experience on the South-North Atlantic Transect and the science and the people that brought it all together. It was an amazing scientific journey, first and foremost. And I had the opportunity to join this training during the beginning of my PhD, which was about 2 years ago. And yes, it has contributed to my scientific aspirations, but I also want to show you today how much it has influenced more than that and has impacted how I view outreach. And by the way, this is Peter Kroot, who I met, like Smadel said, I met during this cruise, and he has joined me to become my co-advisor with Danny. And it's really a great pleasure. So I would like to start and tell you about the amount of emails that we get from the secretary. And sometimes it seems like it can be a lot. We do get a lot of emails, and sometimes it's very targeted emails, and sometimes it's a lot of forwarded emails. But just so you know, there's a lot of little treasures within the amounts of emails that we got. And a few years ago, something came out through that about the shipboard training exercise, and I read through it and I was kind of saying, well, this is too good to be true. They're offering full scholarship for a month cruise across the Atlantic Ocean They're going to be teaching us all these different skill sets. And I really thought it was too good of an experience to be true. But I also said to myself, I have nothing to lose and I have so much to gain. And I did apply for it in the end. And this was the outcome. It was 25 scholars from all over the world. There was over, from what I remember, 800 applications. And it was really this one worldwide encounter on one ship. The South North Atlantic Training Transect is a training survey that brings together both international students as a collaboration between the Alfred Wagner Institute, the Atlantos, which is the Atlantic Ocean Observing System, POGO, which is Partnership Observation for the Global Ocean, and it's also funded by the Nippon Foundation. The target of this cruise is basically to give students and the scholars the opportunity to learn about the ocean atmosphere and climate interactions in order to give tools in order to understand climate change that's happening now and that will continue happening in the future. So this was a major goal. So this is the beautiful RV Polarstern. For the Germans in the crowd, it's Polarstern, and they get very upset when you don't pronounce it correctly. It's a beautiful, amazing icebreaker ship that's usually or in the Arctic or in the Antarctic Ocean. So they do a lot of this, these field experiments. It's basically a ship that gets stuck in the ice and they use this as a field base in order to do a lot of experiments. It's a very beautiful ship. It's 120 meters long. It can go a steady 15.5 knots in ice of up to 1.5 meters. I think it could go 5 knots. And it can actually break ice that's at least 3 meters thick. So it's a really amazing ship. It holds 44 crew members constantly, and it can hold up to 55 scientists. And just for a fun fact, there's actually 2 helicopters that are always on board. And so that's really cool to see. And what's interesting about the ship is it's because it's between the Arctic and the Antarctic, this program takes advantage of the fact that it is empty between moving from the Arctic to the Antarctic and vice versa. And so they use it as an opportunity to have what we call a floating summer school, shipboard training. And that was what the program that they offered. And they do this with different ships from all over the world. They're offering, if they can find empty berths, for this program. And so I don't think it showed the video, but it was supposed to show a bit about the ship itself. We started in the beautiful south of the Falkland Islands, and we made ourselves up north across the Atlantic Ocean until we reached Bremerhaven in Germany. We reached 10 different points where we got to stop, and on a ship like this, you learn very, very quickly how each minute that you stop, how precious those minutes are. And so we were only able to afford these 10 stops. And during these 10 stops, we collected data. These data points are transects that have been done in the past, and they will be continue to be done in the future, and therefore it can provide for a larger data set. And along the way, we were exposed to different fields, really amazing the amount of knowledge that was on board. We focused on a few main modules. We really wanted to look at climate change, which was led by Professor Peter Lemke, who is a leading expert in the field of climate change. And it was an honor to learn from him. He had so much input to give. We did oceanography, remote sensing, data crunching, marine technologies, and outreach. We even got to release a few Argo floats, which was great to see. And all this data contributes to the larger understanding of what's happening in the marine environment. We also got to enjoy just, the pleasures of being on a ship in the middle of the ocean. It's quiet, it's beautiful. It's only, I think when we were in the middle of the Atlantic, we would see a ship maybe every other day. So it's really quiet, it's really beautiful. We even got to have a surprise collection of sediments in front of Helgoland. Like I said, ship time is very expensive, so to make these surprise stops, it's not easy. And this is the fruits of our labor. We were able, not all the data was analyzed on the ship itself. A lot of it was sent back to Helgoland with the Alfred Wagner Institute. But what we were able to do is kind of summarize this experience. So we were able to kind of show what happened in the south, what's happening in the north, in the water, at the surface, and even in the atmosphere. But what I really took away from this cruise was how important, how much emphasis was placed on outreach. And I don't think that's a very obvious thing to do. There's, you're on the ship, the ship is supposed to be for scientific purposes, and yet so much time, effort, and money was allocated just to create Skype connections in the middle of the ocean. So this is actually one of those corny videos that you see when you have no other tools available to you in the middle of the ship. That shows a bit about the outreach that we did, and I hope it works.
Hey, I'm Lisa, and I want to know if you do scuba diving for your projects.
I do a lot of diving in the project that I'm involved in.
I also do cold water diving in Patagonia, and as a biologist, I absolutely love it.
So to answer your question, I think it's very important that we firstly demand political action, and secondly, as individuals, the change should come from us.
What we put up behind you, Marissa's answer.
Oceanography is the study of currents and the overall circulation of water on Earth.
It's absolutely critical that we study the ocean, particularly at this time with climate change going on. And it's such an important lifeline, providing the oxygen throughout the system to the atmosphere and so on. It's an absolutely critical part of our ecosystems.
It's crucial to educate children about oceans and climate change, so they can be empowered and they can make decisions by their own about social, economic, and environmental issues.
So that's just to show the amount of different outreach activities that we were involved in. We were with 15 different schools from all over the world, from Japan, Brazil. We did a climate conference in Germany, and everyone on the site was also writing, tweeting, Instagramming, Facebooking, blogging. So there's really a lot of different things that you can do with such a program. And coming off the ship, I realized I was looking at my fellow scholars, and I was seeing how much they were actually doing at home, how much outreach they were involved in. A lot of these people are coming from third world countries that are still developing. We're not as accessible as what we have in Israel. And I really wanted to do something more. And I joined WISI. And like Smadal said, WISI is a nonprofit organization working in Eilat to raise awareness of the marine environment and bring by social change. We do a lot of educational activities. All of us are involved in lectures, whether it be in front of students, groups, different centers for diving. And we also have a lot of projects going on. So just to name a few, we have some projects that are going on that has to do with raising awareness to cigarette butt pollution, and just kind of make aware how a single cigarette can actually make a big difference and change those social habits. We're doing an eco-conscious diver program. I am currently leading a small project that I'm doing that has to do with bringing artists and creating a book that would tell people about the marine environment in the Red Sea. And this will be available in different languages, in Hebrew and Arabic, also for us and also for our neighboring countries. We're also involved in a lot of different collaborations, because we have to remember oceans know no boundaries, and it's not just what happens here in Israel, it's also what we do as a community between our neighbors, whether it be north, south, east, west. And basically my take-home message is I look at what we do at the school, and I think to myself, really, how amazing, how cool. Everyone is doing some cool stuff. And these are photos that I took from the most recent updates from the school. And I think I speak for all of us when someone asks, oh, what do you do? and you say, well, I'm in marine sciences at the University of Haifa, you get flooded with questions. It always happens to me. I'm sure it happens to you. All of a sudden they want to ask more. So there's a lot of interest. I think it's about us going out there. I think a lot of times we also see maybe more outreach coming out from the PI side, but I want to remind everyone that all of us can do something in outreach. I would encourage anyone who has any special skills, whether it be graphics or if you're able to take wonderful photos or tell stories like Hadal can tell these beautiful stories, to take advantage of them and use it in order to be able to tell more about the marine environment and advocate for what we do, because I think we have a very beautiful world and it's worth sharing. And also, sorry, and I was asked also to tell about opportunities for shipboard training. All emails that I get through POGO, since I'm now a POGO alumni, I send to the school. And a few emails have already been sent out through Einat in the past about shipboard training. If anyone else wants the link, I will post it in the chat below. And I will give you guys an opportunity. Hopefully it's still open if it is. Yeah.
Thank you so much, Debbie. That was inspiring and beautiful. And now I want to jump on a ship myself, which is really rare for me. You must understand. Anyone want to ask Debbie some question? Well, I think it was very clear, very well explained.
Can I comment? Can I say
Sure,
something?
sure, Anna, please.
So Debbie, thank you a lot. And it's actually so makes me feel proud to have you on board the team. And I think really for any of the students who haven't been out on an oceanographic trip, and there are so many opportunities to piggyback on ships. This is a great one, but there are different opportunities, and it's really worth your while to take that opportunity and experience it, because it's an amazing addition to any marine scientist wherever. Even those that you usually don't go out on ships like Smadal.
I want to go. Now I want to go. I'm convinced. Debbie had influenced me. It's not the first time that Debbie had influenced us. Thank you so much, Debbie. And of course, share all the information you mentioned. It will be. I would also.
Can I ask a quick question?
Quick, yes.
Quick. Okay. Hi, Debbie. Thanks for the great talk. You speak a little bit about outreach. If you can, please explain to us what do you think the value of outreach is for scientists, and what is some of the best ways to outreach?
That's a huge question, Madame. A huge question. But I
think it's
very briefly.
Yeah, it's extremely. First of all, for our personal sake, it's extremely beneficial for how we feel. And scientifically, although we don't put enough emphasis on outreach, I really do think that in addition to our academic achievements, our social achievements when it comes to outreach also needs to be a big priority. And there's so many different opportunities. We can talk more about that afterwards. Anyone's welcome. There's just so many different ways you can do outreach. I'm a product of outreach. I was a volunteer at an aquarium, and that's how I got into the field. So if that's not an example, I don't know what is.
Excellent. Thank you very much, Debbie. I think we have more about it in the next two talks. So I want to, I think I'm sharing the right screen. Oh, okay. Yeah, just a second. Okay, thank you, Debbie, very much. Our next speaker is Gina. Gina, you there, right? You here? Gina?
Here.
I don't see you. Can you talk?
I'm here. I'm just outside, and I only have a little tiny light.
Okay. Okay. So our next speaker is Dr. Gina Drake. She is a Zuckerman postdoctoral scholar that is working with Tally Mass. Gina was born in Iowa. You see here, which is a corn-covered state smack in the middle of the U.S., as she herself describes it. Gina didn't see the ocean until she was 12, but once she got in it, she couldn't leave it. her 6th grade English teacher told her that a girl from Iowa couldn't be a marine biologist, but I think Gina had proven her wrong by now. Gina earned her Bachelor of Science in Marine Sciences and Biology at the University of Miami. She's going to be travel a lot, I promise you. After that, she served 2 years as a marine conservation volunteer at in the U.S. with the U.S. Peace Corps in the Republic of Vanuatu. These are a group of small islands that you see here.
Vanuatu.
Vanuatu. Okay.
Vanuatu.
Vanuatu. She worked with the community to conserve marine resources and helped to set up the National Reef Check program. She came back to the US and did her master's in biology at the San Francisco State University. She did her PhD at Rutgers University in Paul Falkowski lab, where she studied the effect of ocean acidification on coral calcification, with emphasis on the coral proteins. And this is where she met Sally. Gina moved to Los Angeles and joined Sally's lab as a Zuckerman fellow, where she studied the calcification protein across several Cnidarian taxa. She's joining us from Los Angeles, where the time is about 4:38 a.m. So I really appreciate her dedication to the seminar. And she will tell us about her studies of fossil proteomics and an after-school program that she led in the public high school. So Gina, the stage is yours.
Thank you. Hi, everyone. I have a little bit of light here. I'm going to wave at you, and then I'm going to turn off my screen so I can sit down and cover up under a blanket because it's cold outside here.
It's nice to see your face, Gina. It's not the same.
I'm sitting outside because my house is small because housing in Los Angeles is quite expensive and my family is still asleep. Let's see. So can everyone see my PowerPoint slide?
Yes, Gina, can you please open your camera so we see you?
Let's see. I don't know how much you'll be able to see of me with a little bit of light outside.
It's okay. It's better than nothing.
Okay.
Go ahead.
There are also crazy people on motorcycles riding at 4:30 in the morning, and planes have started flying over. So I will do my best because, you know, it's Los Angeles and we have planes at 4:30 in the morning. Let's see here. So thank you, Smadar, for including me in this. I've been watching some of the recordings after the fact, obviously, when many of you are asleep. And sometimes it's been the big happy point of my day to see all of the science and how you've incorporated outreach into that. So thank you to the school and Smadar for organizing this. So as Smadar mentioned, I am a postdoctoral researcher with Tali Maas's group. I'm a Zuckerman STEM Leadership postdoctoral fellow in the Marine Biology Department, and I work on coral biomineralization broadly defined. Both the fossil proteomics and the beginning of the after-school proteomics program that I'll talk about today, they were started during my first National Science Foundation funded postdoctoral fellowship. The main outcome of the fossil coral proteomics was recently published, and the after-school program continue remotely.
Gina, can you go into presenter mode so we see the slide more clearly?
I guess for whatever reason, when I do that, I can't see any notes.
Oh.
Is it okay if I use it this way?
I think it's okay.
Okay, sorry. So as I'm sure many of you have heard from Tali and members of her group, we work on stony coral biomineralization. And stony corals are those mostly colonial cnidarians that are composed of 4 cell layers, and over geologic time, they can build massive reefs. Coral reefs disproportionately contribute to marine biodiversity, given their relatively small areal extent around the globe, and they provide important ecosystem services such as like wave dissipation and hence coastal protection, and they've historically been the predominant protein source for millions of people. An integral part of the Coral reef structure is the animal itself, shown here, which an integral part of that is the reef itself, the skeleton, that provides an attachment site to the substrate for the animal and some degree of protection. It may be involved in light enhancement toward the base of the animal. The skeleton that the animal produces beneath itself is not merely a geological manifestation of seawater conditions promoting inorganic precipitation of calcium carbonate. It's instead actively biologically controlled by the animal, shown here. The animal layer closest to the skeleton is called the Calicoblastic Ectoderm, shown here in orange, or shown here in this thin cell layer line. This calicoblastic layer secretes proteins that are retained within the aragonite skeleton. And we know this because of antibodies that have been raised against the protein complex extracted from the skeleton that only bind to the calicoblastic layer, shown here in this kind of lighted up portion. When Tali was a postdoc at Rutgers University, she showed that antibodies raised against an individual skeletal protein, in this case, one called coral acid-rich protein 4. So anywhere where you see brown is where that antibody has bound to a tissue layer. This coral acid-rich protein 4 is a highly acidic protein. It binds to the calicoblastic layer preferentially. And we also previously showed using recombinant highly acidic proteins, including this coral acid-rich protein or CARP4, that they can precipitate calcium carbonate. Let's see. So seen here, let's see, make sure that's. So all of this is calcium carbonate that's just precipitated from artificial seawater from a recombinant version of these coral acid-rich proteins. And they can do this at both ambient seawater pH and at seawater pH that's predicted for the year 2100 due to lower ocean pH due to CO2 emissions, leading to ocean acidification. Therefore, if we want to know how corals make these massive reef structures, and if and how they'll continue to calcify when faced with ocean acidification, we have to know what these acidic proteins are and what they'll do, and what they do. Okay, so that's why we want to know about modern coral proteins. But why do we care about really old coral proteins? Well, first, we can develop phylogenetic trees based on gene orthology analysis, which one of Tali's PhD students has recently done with Asaf Malik. This was Tal Zakin's work, which was just accepted for publication at the end of last week. Yay, Tal. We can layer onto these phylogenetic analyses molecular clock analyses based on certain assumptions. But like, what if major climatic events in the past disrupted continuous evolution, which is often assumed in some of these molecular clock analyses? We could use some of these protein sequences potentially to ground truth some of the dating, some of the molecular clock analyses on these phylogenetic trees. Normally this would be done with DNA sequences, but the limit of sequenceable DNA is on the order of hundreds of thousands of years. So what if we could push this back potentially with proteins? Well, proteins from Neanderthals and here woolly mammoths have been sequenced approaching the 100,000 year limit, and that's pretty cool. But fossil proteomics has pushed, in some taxa, has pushed this time limit back over 2 orders of magnitude. So Tyrannosaurus rex collagen sequences that are over 65 million years old have been successfully sequenced. Not the first sequences, those were chickens, but later sequences have been correctly attributed to T-Rex. And this is a hadrosaur from about 80 million years ago. Proteomics has shown that they're more closely related to birds than to crocodilians, which was previously proposed based on morphology. So fossil proteomics can push back the limits of evolutionarily informative biomolecules beyond what is currently achievable with DNA. So these are obviously vertebrates, and corals are invertebrates, and it's much harder with invertebrates to do paleoproteomics because of the low amount of biomolecules in the starting mineral. But it's not impossible. So here is some work from a 66 million year old mollusk. And this peak right here shows the carbon-oxygen double bond, which is indicative of intact peptides, not necessarily proteins, but at least peptides, to suggest that there is some peptide information that's available to proteomics. And 2 years ago now, a 6,000-year-old mollusk had some peptide sequences that were sequenced. So my NSF-funded postdoc, my first postdoc, sought to push back the age limit for invertebrate paleoproteomics. So our work on selecting the most promising specimens and then sequencing fossil proteins used coral fragments from the Los Angeles Natural History Museum's Invertebrate Paleontology collection. I examined a group of coral heads from the Key Largo Formation, so most likely Pleistocene stage 5E, or about 130,000 years old for mineral preservation. I should say I'm grateful to Austin Hendy at the museum. He trusted me to destructively sample 1 to 2 gram portions of the specimens, which is not insignificant. So in the interest of time, I won't go into great detail, but representing the extremes in preservation, I examined an Orbicella annularis that was about 90% aragonite. So this, these two photographs on the left. And that trace elements suggest as primary aragonite. So here's the X-ray diffraction, the X-ray diffractogram showed that it's mostly aragonite, and then its trace elements right here showing that it's mostly primary aragonite, or that which was originally laid down by the animal rather than secondary aragonite that would have been recrystallized with likely subsequent loss of endogenous organic matter and or gain of contaminating organic matter. I also examined a Montastria annularis that was about 3/4 aragonite and about 1/4 calcite, and then another Orbicella annularis that had been completely recrystallized to 100% calcite, as shown here by the X-ray diffractogram, as well as the trace element data. I'm not going to show it here, but I also examined amino acid racemization, or the conversion of amino acids from the L form that's originally made by the coral animal to the D form, and this conversion occurs naturally over time. And I also looked at the relative amino acid composition for signatures of endogenous amino acids in intact peptides. After extensively cleaning the finely ground skeleton from each of these 3 corals, I decalcified them and then precipitated the proteins using acetone. So here we have lane 2, which shows a protein gel of the best preserved specimen, the Orbicella annularis here, that's 90% or greater aragonite. There's at least one protein band, as shown here by the upper yellow arrow. But much of the protein fraction is found in the lower molecular weight peptides that are likely degradation projects of the original larger proteins. Lane 3 shows the partially recrystallized Montastria, and lane 4 shows the fully recrystallized Orbicella. And they also show extensive protein degradation. So I used dual enzyme digestion and mass spectrometry sequencing with the mass spec data compared to multiple protein prediction data sets from family Merulinidae. And the sequenceable proteins were only detected in the best preserved specimen, this photograph here, and from lane 2. 6 proteins were sequenced from this specimen. 3 of them are known from previous sequencing efforts in modern corals, and one of them is an acidic protein in the CARP4, sometimes called SARP1. That means things to a very few number of you, but it's a scleractinian or stony coral specific gene branch of skeletal proteins. So that was kind of cool. But also as part of my first postdoctoral experience, I sequenced some modern stony corals, which Tal, I still owe you those data, including this solitary fungid here and 2 colonial corals. At the same time, I was thinking about an outreach program that could continue beyond that first postdoc and be incorporated into future grant proposals. So I got in touch with a group of high school students at Reseda Charter High School here in Los Angeles. And I thought that the students could learn a really cool skill set that most undergraduates don't even know how to do this kind of work, and that would look really good on their resume. And I would get some help with a massive data dump. In the summer of 2017, I started working with a biology teacher at Reseda Charter High School in Los Angeles to recruit a group of high-achieving students to learn the proteomics software that I was using. 90% of the students at Reseda High School identify as Hispanic or 2 or more races, which in the US is one way that we it's one metric of racial diversity. And so many students at Reseda High School qualify for free or reduced lunches that all students are given free breakfast, lunch, and an after-school snack kit to eat on campus. So these are not, the pool from which these students are drawn is not necessarily an affluent pool. So I originally taught the students how to use this proprietary software called Mascot that puts together mass spec data and predicted protein sequences. And I also taught them how to use NCBI's blasting tool. We're a pretty small group early on, definitely 5 to 10 students. 6 of the students shown in this figure or this picture, which they gave to me as a Christmas present one year. 6 of the students have continued with the program since its inception. The one student that left, his family moved out of the US. And the 6 of the students that have continued are applying to college this year, so I'm very excited for them. In the, excuse me, in the second year of the program,
Gina, you are muted.
Yes, sorry, I had to cough. In the second year of the program, we added 3 or 4 more students. And in 2019, I guided 2 groups of female students in the program to write small research grants to the Archer STEM School for Girls, which is a school in Los Angeles that predominantly serves a white American for many generations and middle to upper class population. And the girls in the research groups that I worked with are definitely not that. 2 of the 6 girls are from families where they immigrated to the US as children. 5 of the girls in the group speak a language other than English at home. It's been kind of fun to see their text messages with their parents as they've been in the program. So I appreciate that the Archer School is making access to these research funds possible for these students. They've definitely had an experience that they otherwise wouldn't have had. Several of the students have to travel long distance, and this isn't just the students that applied for the Archer STEM grant, but across the proteomics program, many of the students have to travel long distances by bus. I live in the San Fernando Valley, and it can take, on a good day, an hour to an hour and a half to get from the far side of, like the east side of the valley to the school. And they come to Reseda High School because there are these specialty STEM programs that they've wanted to enroll in. Others have to be dropped off at the school early or picked up late by various family members to accommodate the entire, excuse me, the entire family schedules. So during the spring of 2020, the 2 groups analyzed proteomic data that I had from a modern coral core that was gifted to me by Lisa Greer at Washington and Lee University in the US. And then the second group looked at protein sequences from modern and fossil fish ear bones, or otoliths, sent from Jarek Stolarski at the Polish Academy of Sciences. Normally the Archer STEM School hosts a symposium at which all grant awardees present their research, and COVID kind of upended all of that, plus everything. But both groups of students I worked with really wanted to display their work somehow, and they ended up preparing and narrating presentations shown here on the right that are on private YouTube links. Both videos are closed captioned, and if you want the links, I'm happy to share those with you, and I have the permission of the students to do so. A cool new development, if we can spin COVID to be cool, to the after-school program as we've adapted to remote work, is that the older students and the newest recruits for the 2020-2021 academic year, they're now learning different types of software to characterize the proteins that the original students from the first year worked on. Over the summer, I taught the students from the first 2 years how to use a couple of programs from XPASI. So people in the biology settings may know ProteParam, ProCite, that characterize amino acid composition and domain prediction. This program HMM-Pred, which predicts transmembrane portions of the proteins, and Signal P, which predicts signal peptides. So the students use these tools to choose like an optimal candidate protein from a set of about 10 protein sequences that I just give to each student. And then in the spring, I'm going to teach them how to model the three-dimensional structure for one of their chosen proteins using a couple of programs like iTASR and PyMol. But the great thing about this year is that the students from the first 2 years of the programs now guide the newer students. We spend an hour together once a week. I do my data analysis on my research, and the mentors guide their newer mentees in their protein analysis, and I just kind of answer whatever questions come up, which actually isn't that often because the mentors are now really well prepared in the various sequence analysis programs. And so the mentors are shown here with the yellow stars. Excuse me, the biology teacher that I mentioned Wendy Wooten with the red star here because she is a rock star. She drops into our 1 hour weekly meetings occasionally to say hi to the students, and she is super proud of what they've achieved. She brags about them to the district, and I'm very proud as well. Another cool thing that's come out of the program is that 2 summers ago, I helped the
I'm really sorry, but the time is up.
Okay, that's fine.
Okay, thank you so much for sharing this with us.
You're welcome.
But we're really, really over time.
Sorry.
It's okay, don't worry about it. It was very interesting, both the science and the outreach. And actually it's interesting because it's a different country and different system. So it was very, very nice of you to share with us. But I'm really sorry because we're past time, and I want to go to our last speaker today. Just a second. Yeah, so our last speaker is Dr. Beverly Goodman Tchernov from the Marine Geoscience Department. Beverly was born in Milwaukee, Wisconsin. She did her she traveled a lot as well, sorry. She did her BA in anthropology at the University of Wisconsin-Madison. While she was an undergrad, she came to Israel for a summer archaeology excavation in Caesarea. This is a kind of outreach activity too. A year later, she returned to work with the dive team and was never allowed to return to the land excavation. She completed a master's in archaeology anthropology at the Penn State University. It doesn't work, just a second. So here, Penn State University, and worked as a contract archaeologist and corporate anthropologist for 2 years. While working in Caesarea, she gained a strong interest in coastal changes and human activities, and realized she had to have a strong foundation in geology. So she did her PhD at the geology program at the McMaster University in Canada, under the supervision of Ed Reinhardt, focusing on ancient Pleo coastline. During her PhD, she continued to spend time each year excavating underwater in Caesarea. She came to Israel for a Fulbright fellowship with Michal Arzi and worked with Yossi Maut from the Maritime Civilization Department from here. She followed it with the postdoctoral research with Amos Agnon in the Red Sea. In 2009, she joined the Department of Marine Geosciences and worked on tsunamis and related subjects in Turkey, Israel, Greece, USA, and Mexico. Today she will tell us about what happens when you are bad at career planning. Obviously, the best can happen when you don't plan ahead. So, Beverly, please, the stage is yours. Beverly, we don't hear you. We really can't hear you. Beverly, you're mute or something. I can't, none of us can hear you.
Okay, can you hear me now?
Yes, yes, yes.
Great, okay, hold on a second. Okay, first of all, thank you, Smadar. Thank you for the invitation to speak today. I confess, and Smadar can confirm, I was phoning her earlier with a stomach ache, not quite sure what to about speaking to you today, because this is kind of, I'm excited about it, but it's a little bit unusual. You know, normally I'm talking more about my research to an audience by way of National Geographic. Rarely am I talking about my relationship with National Geographic as related to my research. So it's a twist that I'm going to try to navigate and see how well I do today. But it's exciting, and it gave me a chance to kind of look over my activities with National Geographic to tell you about National Geographic, tell you about how it sort of interfaced with my career. Because it truly has, and you'll see in a moment, it truly has sort of accompanied my entire professional career. You know, it began from the time I was a postdoc and carried forward to today. So, so I'm going to try to go through and give you sort of little pieces and highlights of some of the things that I've been involved with, and kind of give you a picture, hopefully somewhat into the insights of the halls of National Geographic. So the following slide is kind of a joke. Okay, this is in National little insider National Geographic humor that we always joke that whenever we start a talk, you always put a baby picture or a young picture to trap your audience. so you'll notice if you watch TED Talks or things like that, they're always like, when I was 3, all I wanted to do was be an oceanographer or whatever that is. So here's my when I was 3 photograph when I really wanted to be a race car driver. Nothing to do with the sea. Anyway, so here we go. So how did it begin? Well, the truth to the story is, yes, I did want to be a National Geographic photographer when I was growing up. I think that was probably the dream of half of the people that I've met over the years. And I grew up definitely in a household full of National Geographic magazines. I think that was pretty much, you know, the most important addition to my education growing up. I mean, it certainly was the way that I saw the world. Just like Jean, I like that we have the Midwest very well represented today. I was born in Wisconsin, which isn't too far from Iowa. And I too did not grow up in the sea. And I joke that probably my first visit to the ocean was in my bathtub with a shell, you know, listening to the ocean through the shell in my bathtub. So, you know, you don't have to grow up, you know, with your feet in the water in the sea to necessarily, you know, have that interest. And I think that's where outreach is so important to remember, is that some of the most involved and active sea people didn't start off, you know, in Florida or California. There's quite a few people that were in very landlocked places, and I think that's because of outreach and because of exposure that they get through these different kinds of venues. So my story. So the true story is, I got a phone call on the way up from Eilat, and I remember it especially because, you know, the area where you're driving up and the reception comes in and out. So of course, Dafka, that was the time that I got the phone call from National Geographic asking me questions about my research. So I remember being incredibly frustrated, and finally I said, can you just call me back in about 20 minutes? Is there any chance, you know? And then I hung up the phone, and they did in fact phone me back, you know, 20 minutes later when I was safely, you know, at Beersheba, and I was able to have the conversation. And the conversation was a group, a producer and a team that was coming to Israel who wanted to film Herod's tomb. They had just discovered Herod's tomb. This is Netzer had.
I'm sorry to stop you. Were you meaning to share your slides with us? Because it's only showing the first one.
Oh my goodness. I am sharing it according to.
No, we see only the first slide in the whole of National Geographic.
just a moment. I think when I corrected my mute, it might have. You would think that I've never used this before, but I swear I have.
Okay.
Okay, so all of that babble had nothing to do with what you were looking at. How's that? Is that better?
Can you open it so it will be a presented mode? Because it's very small, or at least enlarge it, because we don't see it well.
In mine, it's on presenter mode.
We see a very small slide in the middle.
Hold on, let me try one more time.
I think you need to press this button.
Hold on a second. One more time. Okay, third time's a charm.
No. Can you press the you are pressing it. You know what you can do? You just enlarge it, like enlarge it so it will show on the entire screen. So that, like Gina did, because it's a very small. It's very hard to see what is.
Yeah, I hear you. I don't know why it's. Hold on a second. Something is either my cursor is on the wrong screen.
The cursor doesn't. Now I see a cursor.
Okay, what are you seeing now?
Yes.
It's the workspace. It's not the presentation.
Hold on. One last try. I'm choosing the PowerPoint. I don't know why it's giving the whole workspace. It shouldn't. Okay.
Beverly, don't choose the PowerPoint, choose the desktop where the presentation will be presented. Okay, the monitor. Don't choose the desktop.
All right. So don't choose the desktop. Don't choose the PowerPoint, you're saying.
Exactly.
Okay, got it.
Even smaller.
How's that?
Oh, yes, excellent. Excellent. You got it. Now you can.
Okay, we're there. Okay, there we go. Okay, so now you're with me. Okay. Okay, so so in this phone call, I'll let this movie play. So in the phone call, just to save time, in the phone call, they basically were coming to do a program on Herod's tomb, and Herod's tomb is a stone box. So they arrived, they went to go film to prepare for this, and they realized that they were really going to have to stretch out the story of Herod's tomb in order to make it interesting. So they started to look at all the different sites of Herod throughout the country. And at that time, I was actually in the middle of my postdoc, and we had published an article in 2006 about the first tsunami finds from Caesarea. And so they began approaching people, and sadly, I joke that I was sort of the last person standing, because we had done the work with Avner Rabban, who had passed away, and Ed Reinhardt was in Canada, so it left me. I was the most convenient person. I was the one who answered the phone, as they say. So sure enough, we spoke, and they were interested in can you hear me? I see Smadar, everything okay? Okay. So basically what happened was they approached me. They asked about filming, including a little tidbit in this special. And they said, can you do a demo for us? And I said, well, listen, to be perfectly honest, it's a pretty big operation. And to do a demo is to do a project. So that's where we began. And they saw I described the work, and they got very excited, underwater, tsunami, etc. So ultimately, what happened, I requested that they fund an expedition. They did, and you can see here in what you're watching, this is actually the coring that we did as the demo. But since I was in the middle of my postdoc, I had plenty of cores in the Mediterranean. I had enough material for years, but I needed cores in the Red Sea. So I even convinced them to do some movie magic. And in the special, we're jumping off the Med-X in the Mediterranean, and we are diving in the Red Sea. So there's a little movie magic in this Nat Geo, because all they were interested in was the technique. And then these cores, here we are pulling it out in the Mediterranean. The cores that we pulled up, I then was able to use for my postdoc. So so it actually funded the work for my postdoc, because when I was doing my postdoc, I was a bit of a lone wolf. There was nobody working on tsunamis in Israel. And while I had wonderful supervisors who were helpful sort of in the, the life sense, I was working on my own as far as the actual work itself and the techniques and everything else. So I was able, because of this funding, that meant that there was no project funding. Nobody had a budget for tsunami research in Israel. So National Geographic essentially provided the money that was required for extending that research. So moving on from here, I hope. Okay, so after this happened, about a year later, I got an email and the subject line said, "Congratulations, you've won." And you know what happens to emails like that? They usually go into your, go into your spam box. This one fortunately did not, although I do know people where it did go into their spam box that year. And I opened it because it said National Geographic in the subject line, and I'd done this with National Geographic, so of course I thought, interesting. And I remember what went through my head was, wow, this is a very sophisticated spam mail, because they've really targeted something that would catch my eye. And as I read it, the email basically said, "Congratulations, you've won. You've been selected as an emerging explorer for 2009, blah, 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 blah." I had no idea what this meant. I had no clue what, where, when. I heard National Geographic. I said, great. I recall I was even quite pregnant at the time, and I'm going upstairs saying, okay, I don't know what this means. I have no idea what this means, but this sounds like a good thing. Well, what it ended up meaning was that I came to National Geographic the following June to join them for what they have a annual get together for sort of their explorers, which is sort of a complicated term, but at that time, it meant the people that were in this emerging explorers and what I call the pantheon. So we're talking about the Bob Ballards. I've put the Titanic in this photo. Bob Ballard, Sylvia Earle, Jane Goodall, these sort of people that throughout the years have been sort of highlighted and profiled and been explorers in residence and sort of the staple people of National Geographic. So you can imagine my excitement when I got to Washington, D.C., and I find myself in a room of 30, 40 people max, most of the time far fewer, including those people. So it was incredibly exciting. And then during that week, in that initiation week, I was basically taught about National Geographic. Now, everyone thinks of National Geographic, of course, as the magazine. And you think of it in terms of television. We were joking about starting this with bum, 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 like everyone recognizes, when Nat Geo is about to be come on. So sure enough, through these connections, I ended up being invited to do a few television programs. We actually have a few in development right now, and I've also done some that have already been produced. The photograph in the middle that I put up here, I put this up there because, believe it or not, the guy with the little GoPro is one of the top underwater photographers of National Geographic. So I always laugh when people feel like, if they don't have the largest, most amazing equipment, top equipment, there's no way it's going to be the quality. So I have this photograph to prove to you. That's the director, who's not the best diver, on the right, and the very, very world-renowned videographer on the left. So in addition to that, they also have a lot of other activities. They now, something new, they have a podcast. So I've had the opportunity to be invited to last year to be on their new podcast. It's only in its 3rd season now, which is very, very exciting. They have mentorship programs. So I was asked to be a mentee of one of the explorers of Sylvia Earle, which she came to Israel a few years ago. This is a photograph from when we were all in Eilat for the Sharks and Rays conference hosted by Morris Kahn. And then later she came and had accepted an honorary PhD at the University of Haifa. So that's been a really exciting relationship to be able to foster. And I also mentor someone myself. So there's sort of an up and coming researcher in South Africa who meets and speaks to me on a regular basis to help where I'm giving, trying to help guide her, and she helps guide me in terms of our careers and future. And then for me, it's been a lot of amazing informal mentoring. So you can imagine going to these spaces and being around people from all different disciplines, and just the conversations and being able to kind of get good guidance for outreach, for how to successfully incorporate these National Geographic activities with an academic life, as well. Now, one of the things that I find to be the most exciting, and people aren't aware of it, is that National Geographic does a lot of work in classrooms. They have magazines for different age groups that are just like they actually have like multiple young people magazines. They have a pre-kindergarten through kindergarten, and then they have, ages 10 to 13. And so I've been profiled a few times in those magazines, which is, for me, one of the most exciting things. I've also given talks in public schools in the United States. So I've been to these kind of off the beaten path type places, outside Flint, Michigan, and, areas where their National Geographic is sending people through their education program to go to these many times lesser served schools as part of their activities. And I'm also connected with their education program for developing educators. So also working with teachers and giving talks in classrooms and answering questions. Now this, for example, Cengage, they actually have textbooks. Okay, so textbooks that are state by state. This is the US and in Canada. So what they do is they take their Explorer work and they put it in as the examples for the education in the textbooks. And for me personally, I have to say that this is perhaps one of the most rewarding things. The idea that a child, a 7-year-old girl is going to read about marine geoarchaeology and have any idea what that is, something that I certainly wouldn't have known about when I was that age. So this to me is, I think it's incredibly exciting and rewarding that they're giving these real life and putting a face on science, a face on researchers who are doing the work for kids to help kids relate. And we do, voiceover things and videos and all kinds of interactive activities with that. And they also, National Geographic also has tours. Okay, they're sort of the not-for-profit side of National Geographic. The not-for-profit is the magazine, the society, and the grants. And then there's the for-profit side. And the for-profit side is actually education. So they do make a lot of money off those textbooks, and they also do tours. So the tours are everything from luxury down to like summer school. Okay, so they have this huge range, and it's similar in some ways to what Debbie was talking about, where they'll also use these ships for science when they can, or to provide opportunities for scientists to go places, and also to invite children, and in particular high school students, to be able to go out to these places when there's berths available or the seasons that are less popular. So a couple of years ago, one of these opportunities took me to the Galapagos, where basically my role was I was an educator on the ship and also had an opportunity to go around for 20 days around the islands and search for tsunami deposits. So, this was a very exciting opportunity. Some people have selfies of photos that they collect. I have photos of myself in front of tsunami signs wherever I go. So this happens to be one from one of the islands in the Galapagos. And I also like to joke that I was in the Galapagos one day longer than Darwin. I don't think I'm going to produce as much, but I did beat him by a day. Okay, so what have the benefits been? It's been great training. It's an incredible venue to share research from. I mean, it amazes me sometimes. There was something on Instagram early this year where I was on Instagram, and it's just ridiculous to imagine, within 2 weeks, 1.5 million people have seen it. So it's an incredibly powerful platform to have your work profiled. It's an incredible place. Okay, for me, it's like, say, finding your tribe. You go there, it's multidisciplinary. You tend to find people, the way that people think, whether you're studying and researching animals or plants or rocks, just the way that people are perceiving things or ideas that are coming out, that you can be in these spaces with people. It's a lot of fun. And of course, being able to share this across that bridge, from the academic, you go to an academic conference and it's amazing and you're exhausted from all the information and from the exciting conversations, but there's, in a way, it's still in a capsule. And so we need these bridges, and National Geographic is one of these bridges that is basically translating that information in a way that, you know, a spoonful of sugar, you know, being able to find the way to communicate it over and make it interesting to those who are not going to be at that conference, at the scientific conference. The question I get a lot: how do I become? How do I get to do this? You know, how do I become a part of National Geographic? My short answer is I have no idea. Okay, you know why? You know how that happens. Of course, be lucky. Okay, but what does lucky really mean? You know, I don't know who gets credited with saying this, but I heard it once and I think it's fantastic. Luck is really preparedness waiting for opportunity. So do what you love. You know, if there's something, you know, if you're loving what you do and it's exciting and you're inspired by it and you're motivated by it, then you're going to be able to communicate that in a way that shows that you're excited about it and passionate about it. And that's what they're looking for. Okay, that's what they're looking for. So it doesn't matter whether, look, I measure sand. I mean, come on, like, you know, this is not a sexy thing. I measure sand. I sometimes look at mud. I mean, that's ridiculous. But somehow, you know, it's not about, I mean, what I'm really doing versus what it means, and being able to take that over and get people excited about it and understand the relevance, and understand the importance in their daily lives is important. Don't run from the camera, okay? This is tough. It's visual, okay? National Geographic is visual. They want to know, what can we see? Can you tell the story of what you're working on? And if you're really bad at this, you won't always be bad at it if you don't run from the camera. You know, just, you need to practice, you need to do it, you need to, you know, it's something that it's natural to be shy about it, but if you practice it, it will get easier, just like anything. It's a skill. It's a skill that you can really improve. So practice if it's something that you'd like to be able to do. Be passionate, I already said this. Apply for grants, check out opportunities. They have training, they have boot camps, they have webinars, they have all kinds of small and big grants for everything from storytelling to training on a camera to, I mean, go look at the website. It's enormous. It's an enormous website. It has lots of things. It's being updated constantly. But if you go in there, you may find you know, little things here and there that you can apply for, that you can sign up for. You know, some things are free, some things are, you know, you pay for, some things you apply for. There's a whole range of things going on that you can do to sort of, you know, if you want them to notice who you are, that's one way to start doing that. And I'm going to end this with ignore the haters. I won't lie. Okay. And this is something with all outreach. Okay. When they put an article in the paper about you, you are putting yourself on in the academic world, you're putting yourself, you know, on the target. Okay. You're putting yourself on the target. Every mistake that's made in there, every mishap, everything they fail to put in there, people will read this and they're going to go, oh, I can't believe they didn't mention so-and-so because this person was so important. And oh, that person thinks that they did everything themselves. Okay, you know what? You're going to have to just accept that, and once you've done it enough, you're going to understand that you have no control over, many times, not none, but many times you do not have control over what happens down the road once you've done something. You can make, and I've learned I can do every effort in the world. I can send the email with a detailed list. It doesn't make a difference. There are certain things that you simply, you're going to have to take the heat and be ready to take the heat, because it does produce academic jealousy. It does produce these other things, and you have to, it's a lot easier to say no and not do it, or you have to, I'm telling you right now, it will happen and it's okay. It's really okay. Now I know we went over time already, so I just want to say that unfortunately in Israel, outreach is not yet on our list of promotional activities. Okay, it's one of these ironic things that the president of the university, usually the upper brass of a university, is very happy about it, because they want to, of course, be profiled. But on the individual, you know, researcher level, it's not something that is going to gain you any points for, you know, being promoted, being you know, it's simply not considered. that's not the case in every country. There are some countries where, like Australia, for example, where they actually give you, just like publications are contribute, every time you're mentioned in the media, it gives you it gives you currency towards your promotion. Okay, so there are some countries where that is already the case, and there are some places where it's becoming the case. They're understanding the importance. It is no longer, in many places, it is less and less acceptable that it is kept within the laboratory, that is kept within the walls of the university. Smadar actually mentioned to me that NSF, the American, American grant agency, they now request that you explicitly describe what you're going to do to have outreach. How are you going to integrate if there's a local community? How are you going to, what are your plans? Do you have any specials? Have you contacted it? Are there any media outlets that are interested, that are set up to profile it? Is it going to get out there? So, I see this as a trend that's going to go forward and become more and more common. I mean, look at the world today with all the, with how communication is. It's, content, and we want the science to become the content, right? We want, the hope is that, let's hope that if we're going to have, what do they call it? Clickbait, that more and more clickbait will be sciences, and it's up to us to also put ourselves out there and to be willing to do it. So I hope everyone, for me, it has been an amazing, I don't know how to describe it, sort of, it has accompanied me through my career, and I don't regret the few papers I probably didn't write because the time that was dedicated over to this side. For me, I feel like as a personally, that my contribution to the world through taking advantage of this opportunity, for me personally, has been just as important, for whatever sacrifice that may mean for promotionable aspects of my resume. But for me, it's really been fulfilling, and one of the things that I'm perhaps most proud of. So I think I'll just leave it there, and thanks for sticking around, those of you who stayed beyond the time.
Thank you, Beverly. That was beautiful. Thank you so much. And Matan, are you still with us? Matan? Is he still with us? Matan? The one that asked?
Yeah.
So did she answer the question?
Sorry?
Did Beverly answer all your questions?
Yeah, but also Debbie's answer was great, because she said she's a product of outreach. But this emphasized more. But still, like maybe we can talk more about what is the best way for outreach, what is the role of social media now, all these kind of things. It's very interesting.
I think, if we ask about what is the best way, I think for everyone it's different. For everyone it's different. I think that it's I think that if you are doing something, part of it is opportunity. Okay, not everyone has the same access. Okay, so if it depends on, if those phone calls happen, then wonderful. My attitude is say yes. If you can say yes, say yes. If you feel you really can't say yes, that it's too hard for you, give them someone else's name. Okay, give them someone else's name that can do it. All right, promote someone else that you feel like that they're a good representative, that there's someone who could don't just hang up the phone. And everyone's the same. Like some people might go to their own elementary school, or maybe where you live, there's something that you would like to some group, some organization that you'd like to help out. I think that there's no too big or too small. I think that any effort is wild, and it's really individual. Do what you're good at. Sylvia Earle would say to me, one thing she used to always say is, do the thing you like doing. If you're a good writer, write about it. If you're a good photographer, photograph. If you're a good dancer, dance about it. Whatever, find the thing, if you're doing the thing that you're most capable of, or working with the people that you feel most connected with, that's how you're going to communicate it in the best way. There is no one right way.
Thanks, Beverly. You made me very happy. I don't know. I feel really, really happy about everything. And this was a beautiful, beautiful seminar. I'm sorry, everyone, and thanks all the 100 people that stuck with us until the end. This is the last seminar of this semester and the last one in this format for this year. In the next semester, we will have scientists who will share with us their science and the things that make them passionate about their work. So it will still be a seminar with a twist. So I want to wish you happy seminar break, and good luck in your exams, and see you all next semester. Goodbye. Thank you so much. You were excellent.
Thank you.
Madame.
Thank you very much.
Bye.
Thank you.
Bye-bye.
Bye.
Thank you. Bye.
Thank you. Thank you very much, Gina, Debbie, and Beverly. It was beautiful.
Bye, Gina. Good night.
Good night.
Bye.
Good morning.
Nice.